TV seminar, which is also sponsored by the Conservation Science. I want to take just a couple of minutes to introduce Doug Futuyma, and then I will turn it over to Doug. So the great thing about Doug Futuyma, and I think this crowd attests to this, is that everybody knows Doug's name. Everybody knows who Doug is, at least at one level.
Almost everybody thinks he's Japanese.
The reason for that is in part because he's a rock star scientist, and in part because he writes and has written for many years the most important and influential books, textbooks, and other books in evolutionary biology, I think, that have been published in the United States. A very kind of brief summary, and one of the many great things about Doug is that his academic history is refreshingly easy to remember. Doug got his PhD at the University of Michigan in Ann Arbor. He went directly to a job at Stony Brook, and he's still there. There was a brief 2-year stint when Doug sort of did a trial run back at the University of Michigan, decided that Ann Arbor wasn't for him after all, moved back to Stony Brook, and that is Doug's academic career. In terms of sort of who Doug is, and at least how I think about Doug Futuyma, Doug's been a friend and colleague of mine for many, many years. We've worked on a number of the main project we've worked on together is being Doug editor and associate editor of annual reviews of ecology and systematics for many years. And so we've worked together for many years in that capacity, although I got to know him way back when I was a grad student. But I think of sort of 3 important things about Doug Futuyma, and my guess is we'll see a bit of all of those in today's seminar. The first is I think of Doug as the most active and I think effective advocate for evolutionary biology, probably practicing in the country today. Doug has been, you could say, enthusiastic, you could say relentless in his advocacy for evolution as an important part of science curricula, an important part of our lives, an important part of just everything in biology for decades. He's written, as I said, the book that at least many people in my generation were completely raised on in terms of textbooks in evolutionary biology. He's written important sort of books about science on trial, policy books, all kinds of stuff about evolutionary biology. And I think his influence has been tremendous as an advocate for evolution. The second is that Doug's a great biologist, a great field biologist. He works on plant-insect interactions and the evolution of Chrysomela beetles, and he's a very accomplished individual working at that interesting interface between ecology and evolution that comprises plant-insect interaction research. The third, which we had a small dose of this morning before coming here, is that Doug's the best burger I know, and I feel like I know some pretty good burgers. And we went out and got Doug a new North American member of his life list, which is not easy off of Playa del Rey. And Doug last night was asking me, so why the hell did anybody come to Los Angeles? This morning I said, you didn't get that movie in New York. So Doug is a very accomplished birder, an amazing birder. He said his life list is over 6,000 out of the 10,000 or so birds of the world as we speak, and going up yearly. And to me, what that says is, both, yes, he's a natural history nerd and loves birds, but also Doug has a very, very deep respect for the role and the importance of natural history and field biology, even as evolutionary biology becomes, overwhelmed with genomics and all kinds of other high-tech stuff. I think Doug will always respected about the sort of primacy of field biology and natural history as a centerpiece of work in evolutionary biology and ecology. Doug's list of sort of accolades and awards and stuff is as long as your arm. I will just, the one I'll mention is that in 2006, he was elected to the U.S. National Academy of Sciences, something which he, I think, deserves. So without saying any more, I'd like to ask you to give Doug a warm welcome.
Well, Brad, you're much too kind, and I greatly appreciate and am slightly embarrassed by that. That very, very. I'm very happy to be here. It's the first time I've spoken, I think, and have both old friends in the audience, or whom I will be talking to today and tomorrow, and new ones to make. And I've already have this morning. The title of this probably should be rather something like Challenges to the Evolutionary Synthesis. And why am I talking about this? I came about 2 years ago to the very reluctant conclusion that my research years are probably pretty much at an end. So I do other things now, like writing textbooks and other kinds of syntheses and doing this, that, and the other thing. So I will include in this talk I'll touch on some of the research projects that I have been on and show you that I think they are relevant to some of the major themes that I want to talk about. I may or may not have time to get through all of my slides, all of my themes, but the general theme is, and I now have to find out how to, there we go, that's how to advance the slides. My general theme is that we all know that the so-called evolutionary synthesis or modern synthesis that took place from the 1930s up to about 1950 sort of established pretty much the framework of thought that we evolutionary biologists today have inherited. And this was a framework based very largely in population genetics, but by no means entirely. And then from that basis, intended to explain the kinds of observations made by people across the spectrum in systematics and paleontology and ecology and morphology and so on. And in particular, the notion that that macroevolutionary phenomena, so-called evolution above the species level, can be explained pretty much entirely in terms of the processes that you can see operating within species. Well, chief among which would be the action of natural selection on random genetic variation, where random means not that any conceivable mutation is possible or likely, but random means random with respect to utility, to whether the organism is in an environment in which this particular mutation would be advantageous or not. As you probably know, that there have been repeated challenges to various components of the modern synthesis ever since, and I have lived through quite a few of those, and some of them are very much with us today. And in particular, there was a period in the late 70s and well into the 1980s in which there was a great deal of ferment. And so, for example, Stephen Jay Gould, one of the prime players in this drama, wrote a particularly provocative essay in paleobiology in, 1980, is a new and general theory of evolution emerging, in which he said pretty much at the end, I can't remember the exact wording, but it was more or less saying that the synthesis is effectively dead. A much more inflammatory publication at the time was this book edited by two developmental biologists called Beyond Neo-Darwinism, which was an eclectic collection of essays ranging from the fairly reasonable to the utterly bizarre by some people, I will not name names, but I just think some of them were slightly like, well, to use John McCain's word, what was his term? Wackos or wacko. But this continues, but usually in somewhat muted form. And so a few years ago, Massimo Pigliucci and Gerd Müller published here the proceedings essentially of a symposium that they called, in which the title was Evolution, the Extended Synthesis, in which, again, the authors ranged from those talking about, for example, the advent of evolutionary genomics and how that very comfortably fits into the framework of thought that we inherit from the synthesis, all the way through to essays saying, well, we need to go beyond that, to some essays that essentially challenge the validity of fundamental aspects of the synthesis. And I hope to talk about one of those later on, and that will be people who claim that a form of Lamarckism should be reintroduced into our thought. Well, as I mentioned, Stephen Jay Gould was a major player in this whole in this for the longest time, and so I want to talk about a couple of the themes that he epitomized and promulgated at great length. He made the claim, and I will right now say that I think that many of Steve Gould's claims were sort of over the top. They were exaggerated, okay? But I think in most of them, there was a real grain of value in what he had to say, and a real grain of truth, and sometimes much more than a grain. So I greatly admire him, while at the same time, I think there are many particular statements and emphases which I think I and most people would disagree with. One of those statements was this claim that the evolutionary synthesis hardened around natural selection of individual organisms as the sort of be-all and end-all of evolutionary process. In his words, organisms became unfettered billiard balls that could be pushed by natural selection in any direction, in effect without constraint. He, on the other hand, defended the notion that there are important developmental and genetic constraints on adaptation. He, in a famous paper, The Spandrels of San Marco, claimed that a lot of features may be non-adaptive features that are byproducts of development. And of course, he most famously, together with Niles Eldredge, introduced the idea that much of evolutionary change is a history of speciation events. That is to say, that phenotypic change, phenotypic evolution, particularly of morphology, was associated with bifurcation of lineages, true speciation, but using the biological species concept. I want to today certainly talk about that latter issue, the issue of punctuation and the evolutionary role of species. I want to say something about genetic constraints on phenotypic evolution, and I hope I'll have time to say something about the contemporary aspects of, or views of developmental biology that are supposed to constitute some challenge to the synthesis, and in particular the question of whether phenotypic plasticity may sort of supplant mutation and selection as the major agent of evolution, and in particular then whether epigenetic inheritance implies a room for Lamarckism. So the first topic is punctuated equilibrium, and the notion here was that Eldredge and Gould claimed that paleontologists are familiar with a pattern in the fossil record which is not one of smooth, gradual, what they called phyletic gradualism, but rather consists of long continued, more or less static morphologies, which then you find abruptly replaced by somewhat different morphologies which remain more or less unchanging thereafter. Mind you, we're not talking here about the sudden origin of new families or classes or phyla. We're talking about the origin of new species that differ in one or a few morphological characteristics, such as the number of rows of lenses in the eye of the particular trilobite that Niles Eldredge studied. Okay, and everyone agrees that that Eldredge and Gould played a very important service by pointing out the existence of stasis, which most people had not known of. Here's a particularly famous example by Steve Stanley of a species of clam, bivalve, that is still living today and shows effectively the same morphology and bears the same name back through at least 17 million years of fossil record. And there's no question that that is something that requires explanation. Well, I also want to point out that many people think that punctuated equilibrium had something to do with the idea of macromutation, which it did not. That's a separate issue, which Gould also flirted with, but they said no, you can have normal, continuous change in quantitative characters. It just happened, like as shown here, you have a range of variation that's, where you get a divergent lineage, but it happens fast. Okay, happens so fast that you usually don't see it in the fossil record. And they proposed that this change that does happen is in a lineage that buds off a static ancestor that remains static, even if we often don't see it. It may be replaced by its descendant or by its sister lineage or its daughter lineage. And they said, they proposed that in effect natural selection was impotent, could not change morphological characters, except in populations that are initiated as small populations and which become reproductively isolated species. They explicitly said that this was an application or an interpretation of the fossil record, specifically in terms of Ernst Mayer's postulate of founder effect or peripatric speciation. Mayer postulated this based on his observation, illustrated most famously by these beautiful paradise kingfishers, in which you have one species, Galatea, which is found throughout the many different habitats on the island of New Guinea, and is relatively uniform, but finds highly divergent populations on outlying islands, such as this absolutely beautiful bird that I'm very fortunate to have seen on that tiny little island there. And so you have different, what he called different species, derivative species, on the islands around there. And Mayer postulated, he said, I can't see any difference in the ecology. I don't know why these different species would have come into existence by pure ecological selection. And so he postulated that there are strong interactions between, among genes which ordinarily tend to stabilize the genome and resist change, okay, because of gene interactions, epistasis. He postulated that if you get a population founded by a few individuals, you will have substantial random changes in allele frequency at some loci, and that those changes constitute an alteration of what he called the genetic environment, which can change the selective value of different alleles at other interacting loci. So this is very much taking the notion that the genetic background at certain loci will influence the selective value of different genotypes at interacting loci. And he said this could then mushroom into an ongoing process that would lead to very rapid evolutionary change. This was interpreted later by population geneticists as implying that you had change by genetic drift from an adaptive peak represented by the ancestral form, going by genetic drift through an adaptive valley at certain loci, and then coming to a new equilibrium, a new adaptive peak. So it would require substantial the action of genetic drift against natural selection. Now, Mayer, of course, Mayer basically learned his genetics from Dobzhansky. And Dobzhansky, in turn, had been very, very greatly influenced by Sewell Wright. And Sewell Wright, as you know, emphasized this strongly interactive view of the genome. And so there was, if you like, a legitimate intellectual pedigree to this particular hypothesis. But it is a hypothesis that, when modeled by population geneticists such as Brian Charlesworth, they found that it really was very unlikely that this particular kind of peak shift would happen in nature unless you had very small populations, so that genetic drift would overwhelm natural selection. And in very small populations, a much more likely outcome would be extinction, or essentially complete homozygosity in a small enough population. And so for a variety of theoretical reasons of that kind, they said that this is exceedingly improbable. And so not only did they reject punctuated equilibrium, or the particular explanation of this pattern that Eldridge and Gould promulgated, but they also basically dismissed peripatric speciation, and it certainly is a hypothesis that has much lower stature now than it did in the 1970s when Eldridge and Gould published. And so, in effect, population geneticists rejected the whole notion that the pattern that Eldridge and Gould saw in the fossil record could be one in which speciation really plays any particular role. Rather, it was much more likely that you simply had evolving populations that are at some optimum for a while, and then the environment changes and there's a new optimum, and you get evolution to a new stable optimum. Well, I felt as if, I'm not sure why I felt this, but I felt, that there isn't necessarily, the last word has not necessarily been said on this, that maybe speciation does play a role, but not in fostering morphological evolution, but not for the reason that Eldredge and Gould postulated. And I came up in 1987 with this short paper in which I suggested that indeed you might have a strong association in which speciation does facilitate morphological evolution for different reasons. And this was based on the following, first of all, on my presumption, whoops, how'd that happen? My presumption that most adaptational changes are associated with shifts to new niches. Think, for example, of the genus Anolis. As a group, they have a really snazzy adaptation for an adaptive zone, arboreal adaptive zone, namely the digital peds. But look at the differences from one species of Anolis to another, as described by Jonathan Losos and other, and those are all adaptations to different ecological niches, okay, different parts of the tree or grassland or whatever. So much of adaptation consists of changes in response to locally prevalent microhabitats or resources. Okay. Secondly, most morphological characteristics are polygenic, and adaptation to a different niche often involves several characteristics that co-act. So you have a complex of co-adapted traits, each of which is likely to be polygenic. Now, if you get hybridization between two populations that differ in a polygenic character, then there can form a hybrid swarm in which, of course, the individual phenotypes that are represented by the two parent types are essentially disappear because of recombination. Okay. And so I said, because of recombination, a new constellation of adaptive characters is likely to be ephemeral in evolutionary time and will not be registered in the fossil record or contribute to long-term evolution. Why? Because such a new polygenic complex may be its spatial spread may be difficult. And of course, if it just remains in a local population of where it originated, it effectively has no evolutionary future. So it has to spread spatially. And secondly, I suggested that the spatial distribution of the particular niche, if you like, the particular microhabitat or resources, is unstable over long periods of geological time. And because you get changes in population structure, and population structure may collapse. Okay, what do I mean by that? Here's the first. Suppose the ancestral type is adapted to the pink habitat, which is patchily distributed. And in a local population with a somewhat different resource, you get the evolution of a new polygenic morphology. Okay? If it's going to have any evolutionary future, it has to become more widespread. But if migrants end up being, for the most part, finding their way not to other patches of that same habitat, but to the more abundant patches of the original habitat, and they simply land there and interbreed with the resident ancestral phenotype, then you will have the loss, the breakdown of the new polygenic character that originated here. Of course, that breakdown would not happen if this new phenotype, this new polygenic genotype with its distinctive adaptive phenotype, if it were reproductively isolated from the pink populations, that wouldn't happen. They'd be able to sort of carry on and make their way through the landscape, and their offspring would, and you would have the potential for spread. Secondly, environments change over time, most dramatically, of course, during the Pleistocene. And what do we know about that? We know that during the Pleistocene, that by and large, adaptation of populations to climatic and habitat change in situ was less common than simply shifting ranges. That is to say, species tend to follow their niches, sort of habitat tracking. And this process is one not simply of the entire distribution of a species shifting to the south and then back up to the north as climates change, but rather it involves the extinction of populations and the colonization of new ones, and those new populations may be founded by immigrants, by colonists from a number of different populations that are their sources. This is a form of gene flow that may be episodic, but may be more important than the sort of ongoing generation by generation so-called trickle gene flow that we're introduced to with most models. So this process involves then the collapse of the original population structure, and again, as you get interbreeding between different types from different populations, they are likely to lose their divergent local adaptations, again, unless they are reproductively isolated from one another. If a reproductively isolated phenotype, which has, let's say a species of insect which has adapted to a new food plant, okay, can follow its host plant around, north and south as climates change, retaining its adaptation, its distinctive adaptation to that plant, if it's reproductively isolated, if it's become a different species. If it hasn't, and it interbreeds with the ancestral type that fed on the ancestral host plant, then you're going to get, I'm claiming here, very likely to get the loss of the adaptation to the novel host plant. And so I suggested then that what you may have going through time is character which adapted to the ancestral habitat remains more or less stable through time, but divergent populations, which may become phenotypically quite different, but ultimately in the course of time are likely to interbreed with the ancestral type and so lose their distinctive character and meld back in to, and be lost to evolution and be lost to paleontologists because their tenure on this earth was too brief and too localized. Okay? Whereas if they had become reproductively isolated, symbolized by this red bar, then they can happily persist on their own particular resource or microhabitat. And so the phenotypic change providing adaptation to this to a new resource is able to persist through time and indeed be the parent perhaps of a clades in which some species by a similar process become more divergent still. Okay? And so this was my suggestion that Gould and Eldred might be describing correctly, conceivably, an association, a causal association, if you like, between morphological evolution and speciation, but for a totally different reason than what they had originally postulated. This idea, this suggestion has found some favor among some biologists, and actually, especially among paleontologists. Gould rather fulsomely treated this idea in his great big book that he published very shortly before his death. Is there any reason to think that there's anything to this? Well, I'm not sure. I haven't thought of any explicit, absolute tests of it. But if it is the case that morphological evolution is associated with speciation, I mean, at least this would, I think, be a plausible hypothesis. There might be others, but a plausible hypothesis to account for such an association. So the question is, do we see any evidence that divergence of phenotypic characters is enhanced by speciation? And the answer is, at least some papers claim yes. And this is true apparently both at the molecular level and at the morphological level. So Mark Pagel and co-authors have written a couple of papers claiming that there are statistical ways in which you can sort of ask how much of the total variation in a clade of organisms in some feature is attributable just to anagenetic change within lineages versus the number of lineages essentially that have evolved. And they claim that for DNA sequences, that speciation is an active force in promoting genetic evolution. And a Swedish biologist named Bokma has again written several papers analyzing the body masses of mammals and claiming the same thing, and saying that this suggests punctuated equilibrium. It's clearly not true for everything. There's a paper on salamanders that says that Dean Adams and co-workers can't find any evidence of it in salamanders. I think this kind of analysis or the kind of statistics that they're using might well be applied to a variety of characters and a variety of taxa. So far, there's been very little investigation of this kind. The basic ideas here were promulgated early on in the 1970s by John Avis and independently by Bob Ricklefs. But this particular approach is both the nature of the data in the case of Avis and the nature of the statistical analysis in the case of Ricklefs were at fault. But this revives the concept that they were already trying to apply. Okay. The most fascinating paper came out just 2 years ago by a student of Steve Arnold's, and as well as a couple of their co-workers, who analyzed the absolute amount of divergence that you see in body mass in mammals as a function of the time interval over which the change in body mass is taking place. And this involved data sort of across the spectrum, both from phylogenetic analyses of living mammals and from the fossil record. And so here you have, expressed in terms of the number of standard deviations, the difference in log size between two mammals as a function of the interval since they diverged from common ancestor. And we know that you get rapid ongoing evolution of body size and everything, many, many characters, if you look at short intervals in the fossil record. Okay? But it turns out that in general it doesn't go anywhere. It doesn't accumulate. Okay? Here they find essentially no increase as you go from 10 years up to 10, let's say here 1,000 years, or to 10,000 years. You see essentially no increase in the amount of divergence accumulating over that time span. Okay. Not until you get up to around 10 to the 6th or more years do you start seeing that the evolution of body mass starts to accumulate, so that older lineages become more and more different the longer the time since they started diverging. But before that, there's a whole lot of kind of spinning wheels without going anywhere. So they call this the million-year burst, or the million-year wait for evolutionary bursts. Okay, and they don't really go into the question of what could be responsible for this. Okay. At least one possibility is you say, well, a million years, that is on the general order of how long it tends to take for speciation to happen, okay, at least in many taxa. And so, I think it is at least a plausible hypothesis to account for these data to suggest that. Otherwise, I think it would be very interesting to ask what hypothesis will account for these data. Why isn't there a effectively linear or more constant accumulation? So that is where I'm going to stop that particular theme. So that's theme 1. Theme 2 is the issue of genetic constraint. And here, so here's Steve Gould again, being provocative again in his last book. Darwinian variation must be copious in amount, lots of genetic variation, small in extent, meaning essentially polygenic, lots of small changes, and effectively isotropic. And that's the billiard ball. The whole idea that the variation is unconstrained in terms of the direction in which features can evolve. And he says, only under these stringent conditions can natural selection be legitimately regarded as creative. Well, I think that's a gross overstatement, but at least, the constraints are worth thinking about. And then, of course, very famously in a paper with Dick Lewontin, essentially developed this argument as a critique of the so-called adaptationist program. And part of this was the notion that constraints of various kinds may also have the consequence that some features of organisms are byproducts of, for example, pleiotropy or developmental processes and are not adaptations in themselves. And that was the nature of their... Now, before going any further, I want to point out that there were people who were major participants in the evolutionary synthesis who very much did think in terms of evolutionary and developmental constraints, and by and large, these were the more organismal people. Okay, and so Ernst Mayr, in his 1963 book, talks about how you have to sort of translate his words into the concepts that we use today. He's saying a well-knit system of canalization tends to narrow evolutionary potential, and it may account for parallel evolution. Okay? That there's certainly certain directions in which a particular lineage, he said, for example, cuckoos. He says there's been the independent evolution of brood parasitism several times among cuckoos. There's a propensity to evolve in a certain direction. Okay. And he thought this had something to do with developmental or what he's calling canalization. And he says which gene will mutate is random, but the subsequent fate of such a mutation is controlled by the gene complex in which it occurs. Okay, again, by its interaction with other genes. You know, Sewell Wright all over again, except Meyer would never admit that. In Ledyard Stebbins in 1950, and talked about this, and then again in 1974 in a book on flowering plants, in which he talked about adaptive modification along the lines of least resistance. He said, suppose there's selection for higher seed production in a plant. How that is achieved depends on the particular lineage of plants you look at. If you're talking about lilies, we know that the number of ovules per carpel is variable, and that's the way in which you would get an increase in seed number. But in buttercups, there's only one ovule per carpel, and so you'd increase seed production by producing more carpels, okay? Which are variable within species of buttercups. But if you go to the sunflower family, the Asteraceae, there's only one ovule per carpel, okay? And there's only one carpel per flower. That is uniform throughout the Asteraceae, and instead what you will get is changes in the number of florets, the small flowers that make up the inflorescence of the Asteraceae. And so that's what he was talking about. On the other hand, it is true that population geneticists tended to be so impressed by the abundance of genetic variation that had been revealed by studies for decades in almost every characteristic that had been studied, that Dick Lewontin wrote in 1974 that there appears to be no character that cannot be selected in Drosophila. There is good reason to suppose that any outbred population will contain enough variation with respect to almost any character to allow effective selection. And here's a whole bunch of other quotations, including, for example, up to Nick Barton and Partridge in 2000, saying much the same thing. From this point of view, of course, one should be tremendously optimistic about the prospects of species in the face of environmental change. Selection should be able to act, have the necessary raw material to work on to enable evolution toward the optimum and persistence. But on the other hand, we're all familiar with phenomena that really seem to contradict that optimistic point of view. Almost every species that has ever existed on Earth is extinct. So the capacity of genetic variation and natural selection to produce adaptation sometimes falls short, often falls short. There is also the issue of character conservatism. One of the things that will always remain vividly in my mind from my course in insect taxonomy is that the fly family Sepsidae, these little flies that look like winged ants that you can find on cow pats and other dung, that the fly family Sepsidae, which I think has at least 1,000 species, is characterized by having a single anteriorly pointed bristle on the posterior margin of the metathoracic spiracle. Okay. Dobzhansky, all 1,000 species over the course of millions and millions of years retain this particular bristle. Dobzhansky was impressed by the fact that all the species of Drosophila that he knew of have a pair of convergent vertical bristles. Again, fly taxonomy is based on the number and position of individual bristles, and the entire hundreds of species of Drosophila have one pair of bristles on the vertex behind the eyes, the back of the head, that converge. They point toward one another. And he said this cannot be an adaptive trait in and of itself. It is somehow being retained because it must be connected to deeper characteristics of the organism, which you might want to call some kind of developmental constraint, or genetic constraint, but they are mysterious. So in my own case, I'm drawn to this because of the well-known phenomenon of phylogenetic niche conservatism, which is really, really beautifully demonstrated by many, many, many groups of herbivorous insects, which have been my research organisms. And so here, for example, a large genus of leaf beetles, Blephorida, that's been studied by Judith Becerra. These are associations which go back many millions of years, at least to the Oligocene, sometimes back to the Cretaceous. not abundant, and there would have been selection for shifting to something else. And here, over the course of 40 million years or so, that's never happened as far as we can tell. So I was interested in this and thinking, well, to me, this bespeaks the notion that there are genetic constraints, that it's not just a matter of natural selection. If that is true, we should be able to find some kind of correspondence between the phylogenetic history of host plant use in a group of insects and whether or not you can find genetic variation in the capacity to use plants other than their normal host plant. You know, they should be able to potentially have the ability to adapt to some, but not others. Okay, and so that was a long project that I embarked on using an obscure genus of leaf beetles, Ophrella, all of which feed as larvae and as adults on plants in the Asteraceae, the sunflower family. Our first step was to try to estimate the phylogeny and consequently reconstruct host use from this using what is now considered very primitive. We ended up having some mitochondrial DNA sequences, and that's it. I'm sorry, that's all there is. I invite you to redo Ophrella using more of the genome. And they feed, and here's our best estimate, and they are connected by arrows to their host plants. And this is a kind of cartoon of the phylogenetic relationships of the host plants, because these are just the particular genera of Asteraceae pulled out of a much, much, much larger, denser tree of genera of plants in this family. Okay, so there are many, 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 many other lineages of plants intercalated among these branches that aren't shown. And this is not, of course, my work. This is Bob Jansen at University of Texas who did this work. And the plants fall into 4 different tribes of the Asteraceae, represented by different shadings. And what you can see is that there's no correspondence really between the phylogenetic structure of the beetles and that of the plants. They did not co-speciate, okay, at all. But what there is phylogenetic conservatism to a considerable extent in the beetles. So by and large, related species of beetles are feeding on plants in the same tribe, represented by the same shading here, with only 4 switches from one tribe to another. For example, right here is a switch. And in this context, then, I was interested in the question. So I said, I wonder whether a given species of Ophrella has the genetic variation that would be needed to adapt to other plants. And I thought, well, there's no point trying them out on ginkgo or pine or something. People would just say, that's stupid, that's really crazy, what are you proving? What I can do is try them out on other members of the Asteraceae rather than their own personal host plant, and ask whether they're more likely to show the potential capacity to adapt to a plant that's closely related to their own host, or which is the host of a closely related beetle species in the same genus, than to a more distantly related plant. Okay? And so that's what we did, using the methods of quantitative genetics, so simply doing crosses in which you pair individual males with a couple of females and look at and test the progeny, so that variance among the progeny of different males will be taken as evidence of additive genetic variance. And I felt in this case, the conservative, the expectation is that they're going to be genetically variable for everything. So any indication, I will accept any hint of genetic variation as something that is expected, and hence is the conservative hypothesis. And so even just variation among progeny from different females, I take as provisionally evidence of genetic variation. And so we tested 4 different species of Ophrella. It was this one, and this one, and this one, and that one, on as many as 4 or 5 different host plants, which are hosts of other members of the genus Ophrella. Okay. And so, for example, Communa here feeds on ragweed, and we tested it on Chrysopsis, which is in a different tribe. It's the host of that closely related species. And we tested it on Artemisia, the host of this species. And we tested it on Iva, which is a plant eaten by this species, and so on. And what did we measure? We measured a bunch of things. What I will show you here is simply the feeding response of both larvae and adults, all put together in one big summary matrix, in which we ask, and so this is asking, how much do you eat? if we take individual larvae, how much do you eat? And if related larvae tend to show a similar amount, amount of consumption, and so in other words, if it's structured by family, then we take that as evidence of genetic variation. And we simply ask here, did we find evidence of genetic variation or did we not? Okay? As a function of whether the plant that we were testing them on, was in the same clade, it was the host of beetles in the same clade, or was the host of species in a more distantly related clade on that beetle phylogeny. And what you see is, first of all, in about half the cases, we could not detect any genetic variation in feeding propensity. In some of those cases, it was because none of them ate anything. They just absolutely refused. Okay, so you might be a threshold character, but in any case, we saw no consumption. In other cases, you would see some consumption, but no evidence that there was any genetic variation in that consumption. And so first of all, I find this given our liberal criterion for genetic variation, I find this a very interesting observation, which is that in about half the cases, we don't find evidence of genetic variation, despite the claim that all characteristics of all organisms are likely to be genetically variable. Okay? So this is, and I found that interesting. Secondly, it does look as if there is a certain, in fact, a significant degree of match between being more likely to display genetic variation in response to plants which are fed on by closely related beetles and are, in fact, closely related plants in most cases. And so there's a certain degree of correspondence between the pattern of genetic variation seen and the actual history of host shifts in this genus, which to me is consonant with the idea that the history has been shaped in part not just by the environmental opportunities or constraints, but has been shaped in part by genetic propensities. There have been a number of other lines of approach on detecting genetic constraints. Among the most famous is Dolch-Schluter's evidence here, which in some ways, however, I think is weaker evidence. There are a number of other cases, for example, beautiful work by Anthony Bradshaw and his associates on metal tolerance in grasses. Everyone knows about rapid evolution of heavy metal tolerance in grasses. What you may not know is that many grass species in Britain have not adapted to copper tailings or zinc tailings, and those species of grasses, when tested in large numbers of seeds, basically none of the seedlings can survive. There's no evidence of genetic variation in those species of grasses which did not evolve metal tolerance. Likewise, there are a number of others. There's a very nice overview of genetic limits or genetic constraints on evolutionary change by Blows and Hoffman in 2005 in Ecology. And then I have myself, just a couple years ago, a paper in Evolution in which I tried to summarize this point of view. So I think genetic constraints are important. On the other hand, I also think the adaptationist program is alive and well and worth pursuing. Because more often than not, if you see a character well, let's put it this way. If you see a characteristic of a species, there's more profit to be gotten from asking why does it have that character than to assume at the outset that it doesn't have any function. And it very usually does have some kind of adaptive value. Here is a lovely example. So this is wild carrot or Queen Anne's lace, European plant widely introduced throughout North America. And Darwin, in The Origin of Species, I didn't know this, never noticed this until I read a paper by John Maynard Smith, in which Maynard Smith says, in The Origin of Species, Darwin describes how in this umbel made up of a few hundred small flowers, there is one or a couple small dark purple or red flowers right in the center. And Darwin can't imagine what function that could possibly serve, and assumed that it was without adaptive value. And Maynard Smith talks about how he was describing this to a group of people, group of students, I think, in which he said, I can't imagine, why it would be there either. It's probably a developmental constraint or something. And he said instantly 2 people proposed adaptive hypotheses. Well, one of those adaptive hypotheses, he doesn't say what they were, but I'll bet one of them was that it may act as a lure for pollinating insects who think that it is a small insect, okay? And lo and behold, a few years ago, here you have a paper in 2009 showing that small beetles come to umbels that have a dark floret more often than they do if you remove the dark fluoride. And secondly, if you plant small beetles on one of these inflorescences, you get more beetles attracted. Okay, so I think it's an adaptation. It looks like clearly I will not have time to pursue my third theme, alas, and I will let me just tell you what it was. The issue is Baldwin effect or genetic assimilation. Okay, which is the whole idea that you can have a phenotypically plastic response to some environmental challenge, and that over the course of time, what was a phenotypic response can be genetically assimilated and become genetically fixed. We know that we associate this with Waddington, but it's an older idea than that. Simpson reviewed this in 1953. And he said, and he was absolutely right, he said this effect is fully plausible under current theories of evolution. It has nothing to do with Lamarckism, okay? He said it's probably occurred, but there's little concrete ground for the view that it is frequent and important, because there were essentially, he was right, there were no observations that needed to be explained by that. And until recently, I would say that was the case. I mean, I've been looking for convincing cases of genetic assimilation in natural populations of organisms for decades, because I'd like to put one in my textbook, and there haven't been any until very recently. A more inflammatory statement on this is by my dear friend and old friend Mary Jane West Eberhard, who in her book on developmental plasticity said, most phenotypic evolution begins with environmentally initiated change. The leading event is phenotypic change with extensive effects on development. Gene frequency change follows as a response to this developmental change. And so in this framework, most adaptive evolution is an accommodation of developmental changes. Genes are followers, not necessarily leaders in phenotypic evolution. And this has become a very common approach, or common theme. I will simply skip through to point out that we know that there are genetic variations in reaction norms. Okay, the difference, here we have a reaction norm which shows phenotypic plasticity. Here's one which shows much less. It's almost canalized. That particular genotype would have an almost unchanged degree, almost no plasticity at all. And here are different populations of common frogs in Europe showing different degrees of plasticity as a function of heterogeneity of environment. So we know that plasticity can evolve. The critical thing is, where does that phenotypic response to an environment come from? How does it happen? Okay? And I think the critical thing is it something that happens purely by chance, or are we looking at something which is a manifestation of a reaction norm, which is shaped by natural selection in the past to a particular constellation of environmental conditions, and the so-called new condition that you look at is simply one of those. Here's a nice example. Here's a case of Daphnia in which the degree of melanin pigmentation in ancestral type populations, there's little pigmentation if there's little ultraviolet, but in a high altitude ultraviolet rich environment, you get a lot of protective melanin. Except now in some montane lakes into which fish have been introduced, fish see melanized Daphnia, there's strong selection against them, and you've had the evolution, you've had, if you like, genetic assimilation of just part of the ancestral adaptive norm. So that's genetic assimilation, but it doesn't account for the evolution of novelty. I think the best illustration here is by a nice paper by Galambor and others, in saying, suppose here was a phenotypic space, and that's where we sit right now, and here's the reaction. These are possible reaction norms in response to environmental conditions. It might be that the new optimum phenotype lies right here, right along an ancestral reaction norm. Okay? Or it might be that this new optimal phenotype is kind of close to the ancestral reaction norm, but a little bit of evolution in that reaction norm will take it there. Or it might be that the ancestral reaction norm looks like this, and it doesn't point anywhere near the new environmental condition. And we need to know, if you're claiming that phenotypic plasticity is a response to a new environment, we need to know which of these is likely to be the case. Okay? And often it isn't. Okay, and so here's here they pointed out that if you look at human populations that are adapted, genetically adapted to high altitude, they have, for example, high affinity of hemoglobin for oxygen. But if you take you or I, if we've grown up, if we're from a lowland population and we are brought to high altitude, in a few days we acclimate, but the acclimated condition actually is maladaptive. It's the opposite. Okay. We have lower affinity, and likewise we get hypertension at high altitude, whereas adapted populations hardly do at all. And so the critical thing is I think that in many cases that we're looking at is a reaction norm that in the past was shaped by natural selection and just happens to be to point toward the new optimum. Anyway, I think I've gone over time. I'm going to stop there. I'm happy to talk about that or epigenetics with anyone. Many people who need here know more about epigenetics than I do. But my take on it is that those are manifestations of phenotypic plasticity shaped by natural selection in the past and do not replace natural selection as an explanation for the origin of new traits. So overall, there have been challenges to the evolutionary synthesis. I think some of these challenges have real content, but by no means overthrow it. I think that in most cases, as I tried to develop with punctuated equilibrium and the association of speciation with morphological change, I think that we can find that the basic ideas of the evolutionary synthesis are not challenged, but we find new ways in which they can be extended or applied. And I will stop there. I'm sorry for running out of time. Over time.
Microbial community and the microbial genome in that organism is having a big influence on its ability to interact with its environment. And do you think that some of what we're missing here is the fact that we're only looking at the manifestation of an entire community interacting with the environment? We're only looking at the big organism, and we're missing a big part of the picture by overlooking the microbial role.
I don't know enough about that. My sense is that so far no one knows enough about that to have an incomplete answer. I think this is the cop-out is always to say, yeah, it's probably partly this and partly that. The truth is always in the middle. But I suspect in this case that's going to prove to be the case, that some organisms are going to be crucial adaptive roles for their symbionts, their microbial symbionts, and in other cases not. And we certainly know of examples like that. There's all this beautiful work with aphids and their various endosymbionts in which to play a major role in how they can feed, what they can feed on, and so forth. And I suspect in other cases that they won't play that role. But it also is going to depend on what you mean by a role. I suspect that the morphology of mammal or insect is by and large not greatly affected by their physiological capacities, their might be, and their occupation of different habitats. So I think it sounds to me, my impression, and I suspect you know much more about this than I do.
Probably not.
My impression is that this is something we're just beginning to have a preliminary of how huge the microbiome is, and experiments to really dissect it and show all the particular elements of the community in the life of those. I think we would just be able to start.
Yeah, because microbes certainly could control a lot of what diets an organism can survive on, and those diets can then drive morphological change as selection acts on the phenotype. But
we're going to need some serious study to get some sense of what parameters are.
talking about the evolution of body size and how long it takes, like the time scales over which the variance increases. I know for mammals and some other taxa that shape of the distributions looks log normal, and so it's not just the variance, but things like skewness and other features of the shape matter too. I was just wondering to what degree do you think the shape of that distribution can tell you about the mechanisms of evolution or available niche space or phenotypic plasticity?
Wait, the shape of frequency distribution?
Yeah, for body size, for the body size distribution.
That picture was not the distribution of body sizes. It was the degree of divergence in body size as a function of time.
Which is a measure of sort of the, that's going to be related to some degree to the width of the body size distribution, right?
You mean within species?
Or across, either one, within one. Yeah.
I'm not so sure.
Like I said,
No, I guess maybe I'm missing.
well, I just think that, like, no, the degree to which you're diverging would affect, I would think, the body size in the population. Or
within the population.
Or across populations.
Well, not necessarily. I mean, not necessarily. For the most part, certainly at the rates at which this phenotype is very dull, it's very likely that the genetic variance is being replenished by mutation as the more or less maintain itself. On top of which, very often you don't get the exhaustion of genetic variation because of selection, probably because of counter selection, probably because of phylogenetic versus those genes that are more important than the effect of the parent being selected. So I wouldn't necessarily think that these. I think there's no reason to doubt, and I don't think the authors there specifically suggest either for the moment that that pattern is explicable in terms of the exhaustion of genetic variation, which I think is the first.
So I want to go back to the sort of moral level about change happening in speciation, and then if the logic of the argument is that if speciation has occurred, then you're not going to sort of lose that adaptive suite of features through hybridization. You're not just going to push back together with an ancestor. And that's fine. So in terms of sort of who survives through the speciation process, then you're going to have the ones that didn't speciate, they're going to push back, and the ones that did speciate, they're going to survive, and so you're going to get a correlation between speciation and change. But what that doesn't address is something you mentioned, which is the likelihood that those little point range species are going to survive for any length of time. I mean, they're still going to have the little tiny ranges, and you.
Why are they in tiny ranges?
Well, they you know, think about that picture that you showed. They spin off and speciate into one little dot, and they're sitting there initially in that one little dot of habitat.
Right, but there is dispersal, right? There is dispersal, and what I was envisioning was the great majority of migrants will end up, in patches in which they may very well still be able to survive with the ancestral habitat and interbreed there if you want. But certainly some of them presumably will end up in other patches of the same kind of habitat. But over the course of time, if they really, I mean, there are a lot of ifs in this model, aren't there? So maybe it's not all that possible. But
so you're saying because you could still, even if they hadn't fully speciated under the biological species concept, so that they could hybridize, those individuals are still spreading out. So you're kind of saying there's a balance between the rate at which you're going to find those big habitat patches
and the fraction of them that succeed in some sense with endogamous mating, as opposed to outcrossing to the ancestral type.
Right.
So this it certainly calls out for mine.
It does.
If it's worth pursuing at all, I think it should be probably enough.
Yeah.
So I have a question about speciation and also the time scale. like, would you consider that the opportunity for reproductive isolation to be one of the major constraints in limiting
An opportunity for isolation,
reproductive isolation?
meaning the length of time available and the conditions
The
that,
conditions
well, the
would be
conditions
that
under.
one would be to hybridization and these
That's
all
right.
integration and
That's
the
right.
sort
Yeah,
of
yeah, yeah.
the merging of the different lineages.
So, well, I mean, yes. I mean, one of the great concerns, I think, about the notion of allopatric speciation as being a predominant mode in animals, one of the considerations has been how long do populations have the luxury of remaining geographically isolated, you know, during which all the genetic changes that incidentally cause reproductive isolation evolve, you know, how long do they need? And, you know, and how does that relate to the scale over which the distribution of environments, the distribution of geographic barriers, and so on persists? And, you know, and I have a sense that this is a real question. And I've never read anything that to me really analyzes this on the basis of any kind of data at all. And I think that, so this is one of the great appeals of sympatric speciation to some people, not necessarily to me. But one of the great appeals of sympatric speciation is the whole notion that is being driven by strong disruptive selection, and there's really strong selection for reproductive isolation, either as a byproduct of this divergent adaptation or in the form of reinforcement of say, sexual isolation to protect these new gene complexes against doing what I'm afraid they might do, which is to meld into one another. Right? So that then does relate to the question, how long does speciation take? And so you look at Coyne and Orr's book on speciation, and they give, you know, and we don't have a firmly documented frequency distribution for time to reproductive isolation. But for many texts, it appears as if it's on the order of 1 million years or more, with certainly some cases very much more rapid speciation.
It seems to me that the type of plasticity that's most likely to lead to the colonization or replication of new environments is behavioral plasticity, like learning to use new food sources and so forth. So I was wondering if you thought that the same argument could be used for genetic assimilation of morphological traits, behavioral traits. You think it's just the ability for the selection of existing behavioral modes of reaction, or would you say that's incompatible?
I don't see any necessary reason for it to be in a different category, but maybe it is. And I guess, I mean, a tremendous amount of that would have to do with you know, what is a particular animal species? I mean, of course, we're just talking animals. What is the nature of learning in a particular animal species? And there are going to be some in which you can say they are capable of learning, but learning a very limited variety of things. I strongly suspect that Tim Burton's digger wasps could learn, you know, local topography in the neighborhood of their burrows, so that they'd be able to find their way back to where they could bring their prey. Spiders, but that's going to be very different from the learning that your average bird, especially something like a crow, is capable of. And so I think it would be really interesting to know whether learned or behaviorally plastic responses tend to be extensions or manifestations of something that's already part of the normal repertoire. Or are they, you know, or can they be different enough to be what you would call new? How the first New Caledonian crows learned to make hooks out of pandanus leaves it boggles the imagination.
Okay. Okay. One more.
Disruptive technology is rare. So in sort of the human examples, coming up with something so new and it's so disruptive is rare, and most things are modifications.
Okay.